of the BioHub. Today, we're delighted to be joined by Rob Ross, who's the CEO of Clasp Therapeutics, a biotech company pioneering precision immunotherapies designed to harness the power of T cells to fight cancer and other serious diseases. Before taking the helm at Clasp, Rob served as CEO at Surface Oncology, leading the company through its acquisition at Coherus. And prior to that, he was the head of oncology at Bluebird Bio, where he helped advance innovative cell and gene therapies, as well as roles at Genentech and Infinity Pharmaceuticals. Trained as a medical oncologist, Rob completed his residency at UCSF and his fellowship at Dana-Farber Cancer Institute, and he also holds his MD from Columbia University. So join us today as we'll talk through the origin story behind Clasp, the differentiation in the pipeline, and his journey from a medical oncologist to CEO. So Rob, thanks so much for coming on. How are you?
Yeah, thanks so much, Tom. It's great to be here. I appreciate you taking the time and getting to know the story.
Yeah, for sure. It's an absolute pleasure to have you on. So look, let's just start off with a bit about your journey through the field. So what inspired you to pursue a career in the biotech space?
Yeah, so it really goes back to my decision to go into medicine in the first place and oncology in the first place. When I was in college, my grandfather was fighting through, ultimately dying of prostate cancer. And I was very close with him. I named my firstborn child after him, and I was very affected by that. At the same time, I found that in college, I loved science, and I really loved the sort of mystery of understanding human biology and the way all of our organs and all of our systems integrated. And I also found that I loved working with people, and so that got me very interested in medicine. I went to medical school, and in medical school, one of my most profound mentors was a medical oncologist. And what I was most influenced by was the way he worked with patients. Interestingly, he was a former Episcopalian priest. So before he was an oncologist, before he was a doctor, he was a priest. And he was just so good at the bedside and so good with patients who were, as you can imagine, dying of awful cancers. And at the same time, he was a basic scientist and did a lot of work on the underpinnings of leukemia and lymphoma. And so I saw this as a great way to integrate my enjoyment of working with people with science. And so I did my medicine residency and then my oncology fellowship, because I found that oncology was at this great nexus of innovative science and helping people in desperate need. Interestingly, between my residency and my fellowship, I had a year. Because my wife's also a physician and had an additional year for her residency. And my attending in the CCU, when I was a third year resident, was a guy named Hal Barron. And Hal happened to be a senior executive at Genentech at the time. And so I went to Hal and said, Hal, I need a job for a year. I need something to do. And I'm really interested in oncology. I'm going to go do an oncology fellowship. And Hal said, well, why don't you come over to Genentech? You can come and work with us and work on this drug, this antibody we have that binds to VEGF that's in late stage clinical trials. And that was the drug that eventually became Avastin. I had a year there and was just overwhelmed by just how much fun it was, how exciting the science was, how great the people were, and just the leverage you had to really help huge numbers of patients. I happened to be there at an incredibly exciting time. The trials that I worked on were ultimately positive, and this was a huge win for the field, and frankly, a huge win for Genentech, and sort of continued on the trajectory that made it a world leader in oncology drug development. So then I went and did my oncology fellowship. I tore myself away, even though I thought I could stay, but I thought I really wanted the oncology training. Did my oncology fellowship and then stayed on faculty at the Farber, seeing patients and doing clinical research, running clinical cancer trials. And I found that I really loved seeing patients, but I really missed the clinical development intellectual challenge that you really saw when I was a Genentech, and I missed that part. And so I was trying to figure out how I could do that yet still be able to see patients. And after thinking about it for a couple years, I decided to move back into industry. And that's when I started at Infinity Pharmaceuticals and small biotech. But I maintained a clinic at the Farber. And I maintained a clinic at the Farber for the next decade. So I'd see patients about once a week. So not a super intense clinic, but I still was able to see patients. Yet was able to work on incredibly interesting and fascinating integrated teams on novel drugs in oncology, trying to do something different, trying to do something that could benefit huge numbers of patients. Eventually, I had to stop seeing patients. Eventually, by the time I got to Bluebird, I was traveling too much and I just couldn't make it work anymore. But by that point, I was, I'd really just, fallen in love with the work of developing drugs in oncology and moving into more of a leadership role at that point. So that was my story. That's how I got there. I do miss seeing patients. I miss that part of my brain. Having said that, I've also now really enjoy the leadership positions and being in charge of groups of people, teams, companies to move these drugs forward. And at the end of the day, I'm in this for the same reason that I got into oncology, for the same reason that I got into medicine, to help people like my grandfather, right? To help people who need innovative treatments. And that's what I feel like I've been able to do.
Very good. So you launched Clasp just over a year ago. Can you talk us through the origin story behind the company and the inspiration behind its founding?
Yeah, happy to. I'd love to take credit for it, but really the inspiration came long before I got involved. This was Bert Vogelstein, Drew Pardal, and their labs at Johns Hopkins. And Bert Vogelstein is one of the fathers of genetics, and also very, very interested in immunity and cancer, along with Drew Pardal, who's obviously one of the fathers of that field. And the underlying question was, how can you use the immune system to fight cancer cells, but in a very precise way, such that you're not at the same time attacking and killing normal cells? So we know that problem, right? That fundamental problem of how do you kill cancer cells and not kill normal cells? That is the primary problem in all of oncology, right? That goes back to the use of, mustard gas and mustard gas derivatives, right? How do we kill cancer cells and not normal cells? And when we're utilizing something as potent as the immune system, that question becomes even more important, because when the immune system attacks normal cells, you can get horrible, terrible toxicity, and unfortunately, sometimes patient deaths, which we've seen in oncology drug development. And so what Bert and Drew focused on was, can we use the specific genetic changes that make a cancer, right? So this is the same thing we talked about with precision oncology with small molecule inhibitors, yet use those to stimulate the immune system. So we know that those genetic drivers are intracellular proteins for the most part, right? This is mutations in P53, mutations in KRAS, mutations in BRAF. These are the who's who of mutations that cause cancer. The problem is that they're intracellular proteins. So how can you get the immune system to attack them if they're intracellular proteins? Well, the immune system has a system for that. That is what we call the major histocompatibility complex, or MHC proteins. These, we call them in humans, we call them the HLA proteins. And the HLA proteins sample the inside of the cell and put it on the outside of the cell. So they are, it's like a chef tasting its food as you're cooking the food, tasting it along the way and seeing, do you need more salt, do you need more pepper? So the HLA system, one of the things it does is it will present these mutated peptides that come from mutated P53, mutated KRAS, mutated BRAF, that just aren't on normal cells, and that will present it on the cell surface. And if you can get the immune system to identify and attack those cells, then you have a beautiful way of avoiding on-target, off-tumor toxicity, because these complexes, the HLA protein complex with the mutant peptide, that complex just does not exist on normal cells. And so that was their idea, and really their breakthrough was demonstrating that we can use antibody-like technology to make these binders that bind the mutant peptide in the context of the HLA. So you need both. You need both the mutation and the HLA to bind. And then we can use that as a way of triggering the T cell to attack the tumor cell. And so the entire company was born of that idea. And we've now shown that you can do this with multiple different mutations. So we've shown we can do it with P53. We're doing the same work in KRAS as well, and multiple different KRAS mutations. And that's the underlying genius here, is developing these off-the-shelf molecules that can distinguish absolutely between a tumor cell and a normal cell based on these underlying mutations that make the cancer a cancer.
Very good. And as CEO of a company that launched just over a year ago, I think building out any early stage company has its challenges. Can you talk about what it takes to build a biotech, and what does it take to make the shift from preclinical to a clinical stage company?
Yeah, it's really interesting, and I've been a part of this now at multiple different biotechs, moving from very early through with 2 of them going public and getting into the clinic, and now with Clasp getting into the clinic. We're very proud that we entered the clinic a few months ago with our first potential therapeutic. It's funny because 50% of it is purely internal, right? 50% of it is setting up the systems and the procedures, making sure you have the right people around the table, working with people that you love working with. And the more you do it, the easier that becomes, because you get to know the people you love working with and the people that you might not, and you do a better job of finding those people. And preparing internally folks for the idea that this is a company that's going to evolve, right? This is a company, we're not going to be discovery stage forever. We're going to be moving into the clinic. That's going to involve some gives and some takes, and laying the groundwork for that, right? And setting out your vision. And for me, my vision, and I think for anyone with a clinical background, your vision is getting these drugs to patients who need them, right? That's where you got to go. And that, it takes time and effort. You get better at it, right, as you do it more. And I think as long as you're authentic, as long as you're straightforward, reasonably transparent, and surrounding yourself with great people, then it's a lot of fun. The other 50% is the external environment, right? And so I've done this in the context of I've been a part of this in the context of an external environment that was, unicorns and rainbows, which is the early days of Bluebird Bio. I've done this during the time of ups and downs, but overall a reasonably positive trajectory for the biotech community, and that's where we were with Surface. And then now at Clasp, where the external environment in the last 18 months has been rough, right? And I think you have to make choices about how you're going to build the company based on the probability of the markets being there. And you may say, look, 5 years ago, we could raise money on an IND filing. 2 years ago, we could raise money on early clinical data. But frankly, now what the market is saying is we want to see more clinical data, reproducible clinical data, before you can raise in the kind of raise you want to do in order to generate real investor return. And you have to be aware of that and cognizant of that. The good news is we have very supportive investors, right? And we're in the clinic, so we're generating the clinical data that we're going to need. But how you build the company is in part dictated by what the external environment looks like, what the M&A environment looks like now, and where you see it going over the next 2 to 3 years.
Yeah, and you mentioned about a successful IND and getting into the clinic. Can you share some of the other recent milestones and achievements that Clasp have achieved?
Yeah, I'd be delighted to. So the biggest achievement was the timing from the A to getting into the clinic was just about a year. So now this is because a lot of the work on this lead program, Class 1025, came from Hopkins. So this is a T cell engager. So this is a molecule that binds the mutation in P53 in the context of HLA-A02, and then binds T cells and makes the T cells kill the tumor cells with that mutation. And this is the first T cell engager ever in this space. So in the mutant P53 space, or really any mutant, any genetic mutation that we are attacking using a T cell engager. That IND was filed in the early part of this year, and we're now enrolling patients throughout the United States. So we're very excited about that, and we executed beautifully on this initial program. The other thing that I'm very proud of that we've been able to do is now take this technology of making these binders, right, which are highly specific and sensitive to these mutations in the context of HLA, and now applying it to our next pipeline program. So applying it to new mutations, including mutations in KRAS. And so our expectation is that our next suite of programs will be focused on KRAS, and those are moving forward quickly, and we expect to have more news around those in the middle of next year.
Exciting. And Clasp are looking to address some of the most significant challenges in oncology. Can you share more about what the company's differentiated approach is as well?
Yeah. So it is we talk about this a lot with investors. We have a dual selection approach. Every patient we treat on clinical trials and eventually commercially will have to have both the mutation and the right HLA. So for Clasp 1025, the mutation is the P53 R175H mutation, right? So they have to have that mutation. In the same way that if you have a EGFR small molecule, you have to have an EGFR mutation. Or if you have, an anti-KIT small molecule, you have to have the KIT translocation, right? You also, though, have to have the right HLA. And so the HLA for the first program for 1025 is HLA-A02. That means every patient who enrolls has to have HLA-A02. And in the US, that's a little under 50% of the population. Actually, it's around 40% of the population. In Europe, in Northern Europe, is a little higher. It's about 50% of the population. And then it changes based on the racial and ethnic makeup of the country you're going after. So obviously, it puts guardrails around the size of the population, right? Because every patient is dual selected. Having said that, with P53, this is still a very large opportunity, both in the US and outside the US. What I love about it, though, is that it means that every patient we treat, even from the earliest part of the phase 1 trial, has the underlying biology to respond because they have both the mutation and the HLA. So this, from a clinical development standpoint, means you start to get actionable clinical data and actionable efficacy data reasonably early in your phase 1. So you don't have to wait until phase 2. You don't have to wait until combinations in order to demonstrate whether or not the drug is working. So for our phase 1, this is a monotherapy dose escalation and a monotherapy expansion in patients that are dual selected, as I described. And we will learn a huge amount about the drug just from the phase 1. And then at the same time, we'll do some combination work, particularly focused on colon cancer combinations, because this mutation is highly prevalent, is most prevalent in patients with colon cancer.
Okay, and you mentioned P53 a couple of times. Can you just talk through what the significance of the P53 mutation is in oncology, and how does Clasp's platform target these mutations, and what could that mean for potential treatments of cancer?
Yeah, so P53 is really interesting. It is, one of the hallmarks of cancer is P53 mutations. There are many different P53 mutations. P53 is the single most highly mutated gene in all of oncology, all right? But there are many different mutations in P53. The R175H mutation that we're targeting is the most common of all of those mutations. We have, in drug development, been trying to target P53 mutations for decades. And for the most part, we've been unable to. This is one of the first, not the first, but one of the first attempts to really try to target the underlying mutated peptide. So the way this works is the R175H mutation, which just means that where there was an arginine in the 175 position, there's now a histidine, right? It's a single amino acid change. And that peptide is now presented on the surface of the cell in the HLA protein. So our approach is not targeting the activity of p53, right? So we're not targeting how p53 works in helping to cause cancer. We're just using it as a flag. We're using it as a flag that's on the cell surface that is saying, hey, this is an abnormal cell. This is a cell that has this mutation. So by definition, this is not a normal cell. And we're using that flag as a way to then get T cells to attack and kill the tumor cell. So it's a very different approach than other people have taken to target p53. The nice thing about it, or one of the nice things about it, is that this p53 mutation is trunkal, meaning it happens very early in the development of cancer. So every tumor cell in the body that comes from this initial mutated cancer with the p53 mutation, they all share the mutation, right? Moreover, the p53 mutation is critical for the cancer to be a cancer. So you can't downregulate, you can't lose the mutation. This means that when we treat a patient, we're treating every cancer cell in the body, not just some of them. And one of the common ways of developing resistance, downregulation of p53, we think there's at least a reasonable chance that that can't happen because p53 is so necessary in the cancer being a cancer.
Fine. And this is just a question back to you, actually. So your background, you went from medical oncologist to CEO. I guess it's a less of a traditional route to get to being a biotech CEO. Can you talk us through that transition, and how did you find it from being so heavy on the clinical side to running the company?
So I've loved it. It's been great, and I have people in this field that I look up to, people like David Schenkein, who come from the same lineage, the same approach. I think where it's really useful is in helping set the vision and the mission for the company, because coming at it from the perspective of having taken care of patients, having enrolled patients on clinical trials, having given them these very toxic medications, having dealt with informed consents, having dealt with toxicities from different investigational drugs, you can, in a very genuine and sincere way, lay out a vision and mission for everybody in the company that feels very genuine and feels very authentic, because it's very close to my heart, but very close to the heart of anybody who spent time taking care of patients. So that's a real benefit. And honestly, 95% of people who work in small biotech, they'll never take care of a patient. And the reason they're there is because they want to feel like they're part of something that's bigger than themselves and their job. And laying that out, coming from a clinical background, is, I think, second nature. The part of it that is intimidating, I think, for a lot of physicians, it certainly was for me, is it's basically the language of business, right? So I don't have a background or training in finance or accounting. The world of investors was one that was very new to me and that I had to learn. But I found that it's not that hard. It just takes time and effort and intellectual curiosity. You got to ask a lot of questions. You got to be okay being wrong. You got to get a lot of advice from a lot of different folks. But there are lots of folks out there who are willing to give you advice, right? And some of it, it's just like, early medical school, in that the concepts aren't hard, but the language can be hard. And so you got to learn the language. And then when you learn the language, you're like, oh, that's what that means? That wasn't that hard to figure out, right? I just didn't know what the word meant, right? And I found that to be very true in my experience. Like, you got to be intellectually curious, you got to be excited to learn, and you got to be willing to tell people that you don't know and you need help, right? But it's not like learning, X-ray crystallography, right? It's much more like, like memorizing the Krebs cycle, right? It seems intimidating, but it's really not. It's just language.
Yeah, good. I love that. And this is a question we always finish with on the BioHub, and I know I wanted to give you a heads up on this. But if you were to have dinner with 3 people, past or present, who would it be and why? It doesn't necessarily need to be biotech innovators, although it can be if you like. But
yeah. So I'm interested in your experience here, because I think many people ask this question are going to revert back to something that happened recently, right? So I was just recently in Paris with my family, with my kids, and I was reading up a lot about Marie Curie and about Louis Pasteur. And Louis Pasteur, in particular, a brilliant scientist, but also one of the fathers of immunology, right? And certainly at Clasp and before at Surface, we're all about immunology. So Louis Pasteur would certainly be one of them. I recently finished reading American Prometheus after seeing the movie Oppenheimer. And it's hard to be a leader of a biotech or a scientific endeavor, and you have to be impressed with Robert Oppenheimer, no matter what you think morally, no matter what you think scientifically. Just the fact that he was able to lead and get all of those folks pointed in the same direction and end up where we ended up is amazing. And I'd love to have dinner with Robert Oppenheimer. And then finally, I would be saying this third person since I was, 8 years old. So I grew up in Maryland. I grew up a huge Orioles fan. And my grandfather's favorite player and my favorite player because of that was Cal Ripken. Still is Cal Ripken, even though he's obviously hasn't been a player for a long time. And so, and it's not just that he was the Iron Man and showed up every day. His consistency, his resilience, and his humanity, I to this day find incredibly compelling. So, Louis Pasteur, Robert Oppenheimer, and Cal Ripken would be the 3 folks that I would imagine having dinner with right now. In a year, that'll probably change. I'll be influenced by some other movie and some other trip. So, but it'll always be Cal Ripken. He'll always be one of them.
Yeah, well, look, for 3 terrific answers there, and certainly I'm very excited to follow Clasp's journey going forward. It sounds like you guys are, doing some exciting stuff at the moment. So, thanks so much for helping us out with this. It was a pleasure to have you on, and looking forward to following you guys even more.
Thanks so much, Tom. I really enjoyed the time.